Ma'am, please start.
Right. Okay. Thank you. So, a very good morning to all the participants of the 21 days winter school, the speaker, and everyone present here in the meeting. So, it is my proud privilege to introduce before you the eminent speaker of the day, Dr. Tridep Kumar Rajkoha. Dr. Rajkoha, he is working as professor and head in the Department of Veterinary Pathology, College of Veterinary Sciences and Animal Husbandry, Central Agriculture University, Seleshi, Aizawl, Mizoram. So I'll be briefly introducing his biodata in front of you. Dr. Rajkoha, he did his BVSc and MVSc degree from Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur, Madhya Pradesh, and he obtained his PhD degree from IVRI Izzatnagar, and he is currently posted in CAU Aizawl as the professor and head. So, in addition to his regular duty of teaching the UG, PG, and PhD students, he has contributed to develop and uplift the department to the level of recognition by ICR accreditation system for starting PhD programs since 2017. And he has completed 4 DBT funded research projects, and currently he is working on one NASF project funded by the ICR. And with these projects, he could establish the cell culture and molecular biology research laboratory in the Department of Veterinary Pathology at Aizawl. And he is a specialist, particularly in the swine and poultry diseases. And he's providing the diagnostic services on livestock and poultry diseases to the farmers of the northeastern states of India. And recently, just in September 2023, he has also been awarded another research project on NDV funded by DBT. And he has very significant research contributions. So I would like that the participants should know, because these are very important ones. He has independently reported the first outbreak of porcine reproductive and respiratory syndrome, that is PRRS, in India in pig population of Mizoram, along with ICR Barapani, Shillong, and Nishad Bhopal in the year 2013. And he has published the research articles on PRRS involving its clinicopathology, molecular epidemiology, isolation, whole genome sequencing, and characterization of the field strain as the highly pathogenic PRRSV of Chinese origin. And he has done extensive work on classical swine fever virus, and he has also worked on porcine circovirus too, and has reported the prevalence of porcine multisystemic wasting syndrome, that is PMWS, the PCV2 associated reproductive failure in pigs in NER India, and reported the PCV2G genotype for the first time in India from pig population of Mizoram. And his team has also reported the first outbreak of African swine fever, the topic of his today's presentation, in pig population of Mizoram during the period April to June 2021, and characterized the field ASFV strain as the genotype 2. And his research on molecular epidemiological studies on poultry diseases has recorded a new variant of class 2 NDV, the genotype 22 from NER India, a novel variant of IBV with wide range of tissue tropism, circulation of very virulent infectious bursal disease virus in chicken population of NER India. And his team has developed single cell clone of PK15 cell line devoid of porcine circovirus in collaboration with National Institute of Animal Biotechnology, Hyderabad. And his team, in collaboration with NIAB Hyderabad, has developed an indirect ELISA antibody detection kit for PRRS under the research project on molecular biological studies on PRRS virus in pig population of NER of India for the development of sustainable diagnostics and vaccine funded by NASF. And currently, he's in the process of technology transfer. Dr. Raj Koha, in addition to all these research accomplishments, he has been a visiting research scholar in the year 2016 at the University of Connecticut, USA, under the Overseas Research Associateship awarded by DBT. And he has also worked as visiting faculty at Chulalongkorn University, Bangkok, Thailand, in the year 2022 under NAHIP. Now, till date, Dr. Raj Koha, he has guided 3 PhD, 14 MVSc students. He has organized 121 days winter school sponsored by the ICR, and he has also hosted the 36th IAVP Congress and National Symposium on Advances in Veterinary Pathology for Fostering One Health, Food Security and Environmental Protection in the year 2019. And so far, he has published 24 research articles bearing NAS rating 6 and above, and total of 83 research articles with NAS rating 4 and above, 3 books, 2 book chapters, 6 laboratory manuals, and 7 training manuals. So with this brief introduction, I would like to invite Dr. Raj Kohar to present his talk on African swine fever in India, challenges ahead. Sir, please.
Thank you so much, Mom. Very good morning to everyone who have joined this session of this training, 21 days training. And at the very outset, I'd like to express my sincere thanks to the organizers for providing this platform to speak on this very important disease, African swine fever. Of course, nowadays, everything, all the informations at our fingertips, we know everything if we wish to know. But then definitely through this lecture, I would mainly like to share my experience, field experiences, what I have seen at field level, how the disease it is happening, and what is going on currently. I will be focusing mainly on that. I hope I'm audible to everyone.
Yes, sir.
Yeah, and the slide is moving.
Yes, sir.
Yeah. So yeah, I would like to start it with this question. What is happening actually with the global pig population? If we look at the statistics, you say if we see the years here, 2019, particularly on 2020, the total population, global pig population has gone down. And of course, it was in 2021 as well. Still it is rising up, but it's not picking up. I mean, it's not that people are not trying to improve on pig industry or trying, because demand is ever increasing only. It is not decreasing. But then why the total population is going down or it is not improving? The same is when we look at our national statistics. Of course, our data are available till 2019 up to 20th livestock census. There also it shows the significant decrease in the population of pig, and I'm afraid when the data from 21st livestock census comes up, it will be lesser than this probably. And one of the major factors people are attributing towards this decrease in global population of the pig is because of this, the disease outbreak like ASF particularly. It is a pandemic now. Of course, the COVID-19 pandemic was, everybody knows about it. It was so much focused because the human population was directly involved. But here the population involved is the swine. So although it is a pandemic now happening in 5 different continents, the pigs are dying. So this is going on. And when we look at this picture, you see the almost 50% of the pig population are being produced here in China. And that particular year, 2018 and 2019-20, the population of pig in China, the 50% pig population of the world has suffered from this disease, and the pig population was devastated there in China. That is one of the major attributes of this decrease in global pig population, and that is how this disease it is important. Now, the disease, even in our country, it has spread almost all the states of our country, and those particularly who are working at field level, they have come across of this disease because It is such a devastating disease. We feel so helpless when the outbreak it is happening. One cannot ignore about it. So we all know that this is a very severe and highly contagious. It's in India, it has entered in 2020. Say by 2023, almost the entire country is having this virus now. The disease, it has spread to throughout the country. So this is a highly contagious hemorrhagic viral disease. All we know, I would not like to give much emphasis on this, I mean, theoretical or textbookish information, but then we all know that this is a very severe, devastating disease and mainly characterized by hemorrhages throughout the visceral different systems of the body in pigs. And disease is such devastating that it has to be notified. It's a notifiable transboundary disease. So briefly, that is it. The African swine fever is. Again, these are the information everyone will get once they search in Google or anywhere. They will get it. But then, this is of course important to know that it comes in the order as puviral, as parviridae, and most importantly that there are 26 different genotypes of this virus. Till 2015, the number of genotypes identified was 22. But then in this coming last 5-6 years, 4 more genotypes have been identified. Why this is important? Because if there are so many variations in the virus, it is really difficult for the researchers to develop a universal vaccine which will be effective against all the genotypes. So it is important we should know that this virus is highly, there are wide range of different genotypes, 26 numbers. And all those genotypes are available in Africa. Genotype 1 and 2, they are found outside Africa. And in India, the genotype which has circulated or still circulating and causing the outbreak is identified as genotype 2. It has also been published from the NISAID. From our research work also, we have identified it as genotype 2. Then the virus, again, these are textbookish information, but then important to know that it's a big virus, of course, if you see the size, very complex one. Well-protected genome inside one outer envelope, nuclear, and this capsid protein, inner envelope. Then you have the nucleoid inside the nucleus, the genomic DNA is present. And importantly, the genome size is very big. Research point of view, it is important to know this information. See, the length is 170 to 194 kilobase pair. It's a big genome, which is having 151 to 167 open reading frames. That means so many these open reading frames will be encoding so many different viral proteins with different functions. So that makes it very complex. So research point of view, this is important to know that the genome is big. There are many hundreds of open reading frames encoding more than hundreds of different viral proteins which are having different functions. So we yet to understand completely about the virus, the function of different viral proteins. So that is how it is becoming difficult to develop a good, universal, effective vaccine against this disease. Field practice point of view, this is important to know that the virus will be neutralized at high temperature, but below 4 degrees. It will be quite resistance will survive for long, particularly in serum. It can remain infectious for 6 years at 5 degrees centigrade. So that's really important because the virus will survive for such a long period and can be a source of outbreaks even after 5-6 years. In lower pH, the virus is easily neutralized. And for these infectants, I mean, the one of the main important control measure for the disease is to mainly the biosecurity measures and disinfecting the premises. So the most easily available common disinfectant which may be used is the formalin fumigation. It will be effective. That is important to know field practice point of view. Then this is of course interesting, very interesting to know how the virus, this ASFV, which has originated actually in 1921 in Kenya, this red spot here in Africa. Here it was first recorded. Then how this transboundary disease, of course, has crossed the international border, then intercontinental border, made a complete round of the globe, has reached to the Asia, India. Now it is also reaching to the North America, which was considered as free from this virus. So it has started from here, but then this has by 1957, from these African countries, it has reached to the Europe, and I think in the Lisbon in Portugal, near an airport, the disease outbreak it occurs, and source was identified later that those food waste from the airlines carried and used nearby the airport for feeding the pigs, that was the source, and that is how disease it has spread. Then from the Portugal, the entire Europe, slowly it has spread in different years. By 1970, this has also reached to the South America. Then for Asian countries, this is one significant event what has that has occurred in 2007 in the disease. It has spread to Georgia, and the dominant genotype strain here is the genotype 2. Then from this point, the disease has spread to the Eastern Europe, Western Europe, and the most important event that is important for us, of course, for the South Asian countries, is the first outbreak in China. It has occurred in, and the source was from this, the 3rd August of 2018. And once it has entered it in China, then it has spread to throughout almost all the Southeast Asian countries. And by 2020, this has entered into India. And how it has begun here in India is at the beginning, I think during February, March, it was observed that the tributaries of the mighty Brahmaputra, which are coming from the Chinese side, we're having some floating carcasses of dead pigs, and along with that, the first disease outbreak, it has occurred in Arunachal, subsequently in Assam, and that is how the disease has reached to the northeast region of India. Then, of course, in 2021, we have reported it from Mizoram. It was a very severe epidemic form of the disease, and probably for Mizoram, the disease entry point was through Myanmar, because in Myanmar, the 2019, the disease outbreak, it occurs. So this is how, you see, from Africa to Europe to Caucasian region, then the China, Southeast Asian region, and has entered into in India. This is the recent status of India. Almost all the states we have reported officially the outbreak of the diseases. These are the states mentioned here. I hope my slides are going smoothly.
Yes, sir. Perfectly
Yeah.
fine, sir.
Okay. So the latest information, 2023, since September, October, you see these points, they are the recent outbreak which happening, and also it is being reported that the wild pig population in these pink points, they are not very distinct. Of course, I took the map from the FAO, their information. So wild pig population in this entire southeast region, the disease has been reported. And why I'm mentioning this is because it is considered that the virus will be maintained by this wild population of pig. If this is so, see this, look at this image of this map showing this red color, bright red and deep red, the predicted dense population of wild pig habitat. And that color we do find here in near the western part of India and the Southeast Asian countries. So I think we can easily imagine, we can guess it, how it is going to happen, what is going to be the challenges for us. Of course, in my topic, the last two words added is challenges ahead. So this is what the main challenge we are going to have in coming days, coming years. Yeah. Coming to the host range, of course, this is host specific disease to the Swedish family only. And of course, in the soft pigs, this Ornithodoros movata, they also carry the virus. They are also the host for the virus. Of course, this warthog, forest hog, they are the African, the pigs, wild pigs. We do not have it here, but we do have the wild pig population here. These are the host range. Then how the disease, it is being cycle transmitted. There are 4 different cycles. One is the sylvatic cycle. This is important in the African countries because these warthog populations are there. So these soft pigs of warthogs, they inhabit in the furrow of these warthogs. The warthog, the adult population, they may have the viremia, but will not have, they are resistant to the clinical disease. They will not suffer from the disease. However, the juvenile young warthogs, they will have febrile condition, some clinical mild diseases. And this is how among the soft pigs and the juvenile warthogs, this continues there in African countries. For us, of course, this domestic pig and ticks, this is yet to be established, but there is every possibility this might be happening. And once the outbreak it occurs, pig to pig, that cycle is going on. If our wild pig population is already infected, carrying the virus, then this cycle will also be important for us here. Then coming to the clinical signs, of course, those who have worked in field, they will know very well what is this. I mean, once the disease is happening, one cannot ignore it. It is, I mean, visible everywhere that this is happening, and it is frightening to see how devastating the disease is. What happens is, what you will see in the pig farm is sudden severe depression. The pigs, all they will undergo depression, will have high fever, they may have difficulty in breathing, and so they'll just stop taking food. And if they're pregnant animals, there will be storm of abortion and death of both the fetuses as well as the sows. That is what it happens. And the mortality, it range from 90 to 100%. Actually, at the beginning, what we have seen is almost 100% mortality in the affected farm. Of course, now I'm coming to the actual original pictures that I have collected from the field outbreaks. This makes the disease more complicated because in naive population, immunologically naive population, the disease, of course, the first outbreaks, it has happened in epidemic form. It was very severe acute form. All the pigs died. But then 2020 to 2023, the virus is slowly establishing the endemic status here in pig population of India as well. And with time, of course, there will be the virus will change, evolve, and there will be the host pathogen relationship. So that is how the disease always evolve clinically as well. So now maybe we'll be getting to see the subacute and chronic form as well. Subacute is somewhat similar to the acute form, but the course of the disease will be long and the mortality may go down 30 to 70%. Chronic disease will be very confusing with PMWS caused by PCV2 and some chronic CSF infection. So it is going to be a complicated one now. So that is it. Now, what actually we have seen at field level, that is what I would like to share now, and I feel that is more important than the other textbookish information which is easily available. So these are the pictures from some of the affected farms. The affected pigs will be lying like this. Will have the high fever, maybe diarrhea. Some will have bloody diarrhea even, and severe depression. And the ultimate result is outcome is this: you will have to dispose the hundred of pigs together, so devastating the disease when it is happening in epidemic form in immunologically naive pigs. So coming to the pathology, clinical aspect, of course, that I have already said. Now, clinically, I mean pathologically, what you will see in dead pigs, being a pathologist, of course, this is very interesting to me. This is one boar died of that first outbreak here in Mizoram. You can see the black spots here on skin. They are not the other paint or something else. Those are the cutaneous hemorrhages. This is more clear in this ventral. You can see two different cutaneous changes here. These are the erythematous, actually like they are like hematomas, and otherwise this is diffuse, severe, the change of color of the skin. This diffuse, the bright, somewhat pinkish discoloration of skin we may see in acute CSF, PRRS. So this may happen, but the black, the patches of hemorrhages, cutaneous hemorrhages, I think this is more clear here in this picture. You can easily see it looks like hematomas, as if there is accumulation of blood beneath the skin. This is how the cutaneous lesion develops in affected pigs. In the first outbreak, we have seen these cutaneous changes. So definitely there are hemorrhages. At the very beginning, I said it's a hemorrhagic viral disease. In different the visceral organs, there will be hemorrhages. So at the time of that, you may find the bleeding through nostril, through anus, even through the vagina. We have seen it that there may be the oozing out of blood in dead pigs. Some little variations in the, see, this is so in some other age groups. See, this is in a grower pig. On the skin of testicles, bright red cutaneous changes. Those, of course, on ventral aspect of the skin, you see the hemorrhages. They definitely look different from which occurs in PRRS, CSF. In CSF, PRRS, it may be a bright red colored petechial type of hemorrhages, but the patches, these black patches of hemorrhages, cutaneous hemorrhages, I have seen only in this epidemic form of the first outbreak of the disease. Definitely, when this type of cutaneous hemorrhages, as soon as you cut the skin, subcutaneous tissues, there will be oozing out of blood from those hemorrhages and all the lymph nodes. That is what one peculiar, unique changes I have seen is that in other diseases, CSF, PRRS, PCV2, some particular regional lymph nodes are more severely affected. Here, we have seen that almost all the regional lymph nodes are very much enlarged, will be dark black color hemorrhagic, and it goes under necrosis as well. So these are the different regional lymph nodes. This is hemorrhages, petechiation on epiglottis tonsils. Then when you open the abdomen, look at the severe hemorrhages here in the large intestine, diffuse hemorrhages on the wall of large intestine. And this mesenteric lymph node, it's as if it is looking like a clots, blood clots. It has severely hemorrhagic and blackish in color. When you cut open this large intestine inside, these are the mucosal surface, it will look like this. You may also see the fresh patches of hemorrhages on this wall of the small intestine. So this kind of severe hemorrhages, it occurs in the epidemic form. And I have also seen the severe hemorrhagic cystitis. In that case, there will be hematuria. That thing also we have seen at field. The liver is severely affected in this disease. You will see that entire liver will have hemorrhages and necrotic white foci. It looks like this. And one of the important lesion of this disease is this gallbladder. It will be distended and there will be hemorrhages on the wall of gallbladder, which is not commonly seen other diseases like PRRS, CSF, PCV2. Then you see this another liver, the hemorrhages on the wall of gallbladder. This, the hepatic lymph nodes, it will be enlarged so much that it becomes like it's bigger than a walnut. See this we have collected. This is the separated one, the hepatic lymph node, the gallbladder with the hemorrhages on surface. The gastrohepatic lymph nodes, these are important findings for making the differential diagnosis of ASF with the other highly pathogenic viral diseases of pigs. So these gastrohepatic lymph nodes also becomes enlarged, severely hemorrhagic, blackish in color. Definitely the size of the spleen it is, there will be splenomegaly, very big, it will cover the entire surface of the abdomen if you spread it. So that thing you will see in ASF. But that, of course, I have seen it in acute case of PRRS as well. So the lungs changes, it happens. Either you will get a diffuse edematous hemorrhagic lungs, but in that also, like the black spots of skin, you will get this ecchymotic type of hemorrhages on surface of the lungs. This is one peculiar changes that we have seen in case of ASF. And you can see the heart here, the severe hemorrhagic myocarditis. So this happens, this I have seen only in ASF, not in other diseases. And there will be severe hemorrhagic endocarditis. The kidneys will show the changes like this. This will definitely confuse us because it is matching Turkey kidney with this, which we see in many cases of classical swine fever. But then this is not a consistent lesion. In some you will get to see this. In some there will be few petechial hemorrhages, but in many other you will see the patches of hemorrhages like this, and the renal, the lymph nodes are also very much enlarged and hemorrhagic. This is another picture of kidney. So it's not a constant picture. It will vary. Then, I said that it's already 3 years now, 2020 to 2023. The virus, it has established here in the peak population. Although officially there might not be, I'm just guessing, or every outbreak might not be reported, but this is my experiences said that this is happening very regularly. The outbreaks, it is going on. At least in my place, I am still working on it, getting the outbreaks regularly. This is the recent one we found. See, the clinical disease, it is what we have observed it is already changing. The disease, it is evolving with time. No cutaneous changes like the earlier epidemic form when it has happened in the naive peak population, but then it is there was breathing problem and some protein discharge from the mouth, nostril as well. When you look for those severe changes, hemorrhagic changes on large intestine, small intestine, it's not clearly visible, it's not happening. But of course, the liver is severely damaged. There will be necrotic areas, and the spleen is also affected. Now, look at this area of the liver, this area, the necrotic area, actually. How does it look like? So severely the livers, it is affected. So then the lungs, of course, similar picture as we have seen earlier, we are getting it. The heart is very, frequently we are getting the severe endocarditis in the heart. This is another case. It is as if this is the subacute case of the disease. This piglet has suffered from more than 3 weeks, and there was not that kind of severe clinical signs symptoms. The piglet died. There was difficulty in breathing, some nasal discharge, and when you look at the viscera, those severe hemorrhagic changes are not visible. We are not getting it. Instead, the spleen, of course, showing. Instead, the intestine, it shows some button ulcers, like as if it is to confuse it, that as if it is a chronic case of CSF. Liver is affected, but no changes in the gallbladder. So that is how the disease, each time it is evolving. The lungs were severely affected, and there were necrotic white foci areas throughout the lungs, clearly saying that the piglet suffered from the disease for a long duration. It has not died suddenly with the acute course of the disease. These lungs, again, it shows this white, the necrotic focal areas. So this subacute form of the disease, it is now coming up. Maybe in coming years we'll get to see the chronic form of the disease, and that is how the disease, it is going to be a very complex one to make a easy diagnosis, differential diagnosis with the other viral diseases. So those are the gross pathologies we have observed here. Coming to the histopathology, it is well known that the virus target the lymphocytes in all the lymphoid organs, these mesenteric lymph nodes. One can easily see that medulla is totally gone. In pigs, of course, outside the medulla, it's all gone, hemorrhagic. In cortex, all the lymphoid follicles are gone. These follicles, you can see only the reticulin mesh. All the lymphocytes are depleted, and there will be the formation of thrombi in microcapillaries everywhere. One of the very interesting finding I have seen in this is that the lymphocytes are gone, but then those affected area is heavily infiltrated with the eosinophilic type of like cells. These cells are clearly having the eosinophilic granules in the cytoplasm, and throughout this parenchyma, these eosinophilic cells were present. In tonsils, similar lesions in the mucosa, the development of microthrombi, and lymphoid depletion. Spleen, the entire parenchyma is gone, necrose. Again, in that area, you will see these eosinophilic cells infiltration throughout the damaged parenchyma of the spleen. Lungs, one can easily, guess it that when the gross lesion is like that, non-collapsing edematous huge lungs, all the air spaces are filled with those serous type of fluid, and so there is the inflammatory infiltration. And this is the picture. In the kidney, those petechiations, they appeared like this in microscopic sections. So these are the histopathological pictures we have seen in affected organ. Now, for making the diagnosis, of course, this is important to know that 0 day to 14th day, this is the phase of viremia. So if we are collecting the sample during this phase, these samples are fit for detection of the viral antigen, the virus, not the antibodies, because antibodies will come up afterwards. So, and this is the phase where both the antibody and the virus, viral antigen may be detected. And after that, it is the antibodies may be presence, antibody. So accordingly, as per these different phases of the infection, one need to select the diagnostic technique, whether to detect, you are selecting the diagnostic technique for detection of the viral antigen or antibody. So this is important. We have confirmed all the outbreaks by doing the real-time PCR. Then we have also done the absolute quantification of the virus in different organs. So what we found is the spleen is always having the maximum viral load, followed by liver. So then these two samples are best for detecting the virus in different samples. Of course, once it is confirmed, we have also tried to classify, characterize the circulating strain. For that, the universally accepted the protocol is this: doing the phylogenetic analysis of this capsid protein P72 gene, the C terminal of this gene. So for genotyping, this phylogenetic analysis, it is recommended. We have done it because the tree is very long. So I have kept it like this, the tree. This all red dots, the sequences of our sequences, and clearly it has been characterized as genotype 2. The circulating ASFB in Mizoram. Of course, this study is from Mizoram. Again, to make it more clear within this group as well, one can be further see the fine characterization of the virus on the basis of phylogenetic analysis of P54 gene and also ORF9. When we did the P54, it exactly looking the same like P72 coming as a genotype 2 group. But when we analyze the ORF9, we found that the circulating strain from NISAT, actually there were 3 sequences of this ORF9 which have been collected from the Arunachal and Assam. Then all these red triangular sequences are our own. So what we have done is we analyzed this gene as well. We found that this group, it is sub-classified into 2 groups. And what we found that there are one difference in the amino acids in the carboxy terminus of this ORF9 gene of our sequences, that is from Mizoram, and the other sequences from the other genotypes, and the Arunachal and Mizoram submitted by the NISAT Bhopal. And from that we found that the source of the virus for Mizoram was definitely, it was not the same, or it has not entered from the Assam border, or Arunachal, definitely, probably it might have entered from other source like that Myanmar, because this amino acid was not found in the sequences from Assam and Arunachal. So that is how we have concluded that the source of the ASFB virus, the entry point, of course, it was not from Assam, from Mizoram, it was probably from the Myanmar. The Nisad Bhupal has further done the whole genome sequencing, and there are 2 whole genome sequences available. So from that analysis, it is this information has come out that the genome size of our genotype 2, which is circulating here, is 190.5 kb base pair, kilobase pair, and there were 219 putative genes in the genome of the ASFB, which is circulating here in India. Then coming to the prevention control, of course, we all know that this national action plan is well placed. I mean, of course, it has been since 2020. Even before this, all the state governments were alerted for this disease since 2019, probably, because the outbreaks were going on there in surrounding Southeast Asian countries. So this action plan is in place. Of course, the implementation is mainly on the state government department, veterinary departments. We know that for controlling, wherever there is the outbreak of the disease, we identify this infected zone up to 1 kilometer radius. Beyond that 9 kilometer, it will be a surveillance zones. Then, of course, beyond that 10 kilometer, it is free zones. So the practice in the infected zone, surveillance zones, they are well given properly in the National Action Plan. I think all the veterinary field officers are having a copy of this National Action Plan, what we need to do. If it is infected zone, complete restriction, complete quarantine, no movement of anything related, associated with that infected farm. Surveillance zone, again, weekly, we need to be very vigilant in that area. If there is any sick pig, then immediately the diagnosis should be done. Then movement of restriction, quarantine, everything is, nothing can get out of this. Of course, the free zone is free from those, but monitoring, continuous monitoring in the free zones. This is known to almost all the field practitioners. Then coming to the challenges ahead, we all know that there is no, medicine to treat it. Then we do not have an effective, universally effective vaccine developed till date. In that case, the future, in coming years, it is only the challenges which I can view, I can see. I mean, we have challenges only, no solution with us yet, until unless the vaccine is developed. People are, the entire scientific community working on ASF, they are working very hard, putting all their effort, all kind of vaccine. You see, whole inactivated vaccine, then the another type is naturally attenuated. People are trying to find out that the isolates from some of the clinically healthy pigs, and they found that those are naturally attenuated, can be used as a vaccine. And people are also trying to attenuate the virus in cell pulses. So that is how the attenuated live vaccine is also, it is being tried again and again. Then also tried for gene deleted live vaccine, then recombinant viral vector, subunits, and DNA vaccines. But so far, of course, there are advantages, disadvantages as per the technological knowledge. Against all these vaccines which are being tried, but so far we do not have the effective vaccine to control and prevent the disease. In that case, we do not have the treatment to treat the animal. We do not have the effective vaccine to vaccinate and contain the disease. In that case, the only the solution remained for control and eradication is the early detection and strict stamping out policy. The early detection also we have problem at field level, because to handle the infected material of this virus, there are strict instructions to be followed, particularly about the biocontainment. We need to have, this is a virus which is put at the risk group of 4, the highest risk group virus. So accordingly, we need the biocontainment facilities of level 3 actually to handle the materials. So in that case, I mean, what say entire northeast region, we have RDDL there at Guwahati. So all the other states will be running from their place to that RDDL for the diagnosis of the disease. So that is how we'll get the rapid detection and diagnosis to adopt the control and preventive measures. I think that's a great challenge for all of us, the veterinarian here. So that is it, rapid detection. Then at the same time, in the State Department laboratories, I mean, how, up to what extent they are being equipped, and they provided the infrastructure to give the rapid diagnosis. You know, there are certain commercially available diagnostic kits. There are some LFA kit to detect the antigen, the virus, but then this disease is highly very complex one. It is found that although I have shown the different zone of the infection, at the early stage, viremic stage, the virus is supposed to be in the serum, but when you test it with that antigen detection LFA, you don't find the virus out there in serum. And they have scientists, they have also, I mean, confirmed it that in some cases the virus, it is multiplying, causing the disease in the lymphoid organs, in the other organs, but the viral load in the serum, in the blood is, they are not finding it. That of course depending on the involvement of the different strain. So we do have challenges in giving the early detection and diagnosis. In that case, I mean, the next procedures to control and containment will totally depend on the early detection diagnosis. We must solve these challenges first. Second, the ultimate solution is strict stamping out policy. I mean, once the farm is having, the virus is detected, it's confirmed, the entire population should be stamped out. How feasible is this at the ground reality at farmers level? If the farmers are asked to kill all the animals, including their healthy animals, and dispose it off, clean it off, who are going to pay the compensation? There are some systems developed for compensation to the farmers, but then from the state government sides, but that compensation is not matching at all with the total value of that pig you are going to cull and dispose it of. A 2 months piglets cost here 7,500 to 8,000. Compensation for all the animals will be given, never more than 5,000 rupees. So in that case, what will be the reaction, response from the stakeholders, the small pig farmers from this country? So these are the huge challenges it is coming up. In addition to this, we do have the porcine circovirus 2, classical swine fever, PRRS, which are the endemic viruses circulating in the pig population here. In that case, when the different highly pathogenic viruses are circulating together, now the ASFB is added to that. There are every chances for syndemic infection of those viruses. In that case, some other challenges will come out, like in that case, what will be the clinical form of the disease, how the animals will suffer clinically, what will be the pathological changes. Those we do not know yet. We'll have to find it out. We'll see it in future. So it is what personally I could see in front of me is only challenges so far, no solution at all. So I think my time is almost, I'm completing my given time. So with this, I am concluding my talk today. I hope I could give at least a few of the um, informations, important information about the disease. So now to
Yes,
the
sir.
audience, please.
Thank you very much, sir. So the lecture was very, very interesting, and it was, I think, an excellent storytelling on African swine fever from your side. And I'm sure the participants, they must have had a great learning experience from this lecture on African swine fever. And particularly means you covered everything, starting from its origin, how it transmitted across the countries, then the continents, and how it reached India, the acute cases, and how the disease has evolved over these last 3 years. And the participants must have also benefited about the diagnostic techniques. You also told about all those things, how you diagnosed them. And in the last, the challenges, of course, they are the major constraint, and means that has to be we look forward to the solutions to this problem. So now I would like, I think there is one question in the chat box. So we can take up some questions. I'll request all the participants that they should not hesitate and ask the speaker of the day with the relevant questions on African swine fever. One question is from Dr. Ankita Modi. She's asking, does African swine fever affect humans when eaten?
No, no, not at all. This is a highly host-specific disease. It will cause disease only in swine, in pigs only. So it's not a zoonotic disease. But then, of course, it's a devastating hemorrhagic disease of pigs. It will not cause disease in human, even if but that will have some other, effect. If it is being used for the table purpose, that will be a very good source of the virus spreading to one place to another place, one farm to another. So that is how actually the virus, it has moved from one continent to another continent, and that is important. It should not be. That is why there is strict instruction that once it happens, it should be killed, culled, and disposed of properly.
Okay, thank you, sir. I'll request the participants, if they have any questions, they should please ask, because the winter school is meant for answering the queries also. The lectures have been specifically designed, and I think this lecture was very good on taking one particular disease, and we have participants from all over the country. So they must have benefited. So there is another question. If the wild pigs are resistant, can